in terms of the clock, let's just start with that. Shinya Yamanaka wound the clock so far back it went back to zero, back to midnight. That we do not want to do because the cells lose their identity, and that's the last thing we want to do. We don't want to go back to a
because it's dangerous to have a pluripotent stem cell in the wrong place in the body?
Of course, it'll grow. It won't stop growing.
Why does it become a tumor? Why doesn't it become a liver or a lung? Like, I would get the problem of having a liver develop in my brain, but I'm just saying, like, why does it become a tumor cell instead of an actual functioning liver?
Well, so when you go back far enough, it loses its identity. It will just multiply into a mass without cellular identity.
So there's something else going on that stops it from figuring itself out.
Right. So in the lab, if we take a pluripotent stem cell and we want to make a liver cell or a neuron, a nerve cell, we give it a bunch of chemical signals in what we call the niche. And when one of these cells lands in a niche around those old cells, they'll use that stem cell to rebuild tissue. But imagine if we could reset the clock, not all the way back to a stem cell, but just partial reset the clock so that you could go back to being 20 again.
Right.
That's what we're able to do in some tissues in the mouse right now.
How do you do it en masse? When it's cell by cell, DNA strand by DNA strand, like, how the hell do you get this to take effect through a whole joint, let alone the whole body?
Right now, the way we do it is we inject a virus called an AAV, and this virus will target certain tissues and deliver the genes to most of the cells in that tissue. For example, we are treating aging of the eye in mice. So we can take an old mouse, we deliver a virus, the AAV, into the eye, tiny little prick. It's the same virus that's used to correct genetic deficiencies in the eye right now, FDA-approved drugs. So this isn't science fiction, this is out there in the world right now. We give it to the old mice, we give them an antibiotic called doxycycline, and that turns on these reprogramming factors. We don't use all 4 of the factors, OSKM they're called, because one of them causes cancer, the M. We leave off the M, we put OS and K into the eye, turn it on, leave it for a few weeks, measure what happens to the eye, and those mice can see again like they were young.